So yes, thanks. I'm going to be talking about cryo-electron tomography today. And as Claudia said, my kind of microscopy history began on the light microscope side of things. And my passion really is around how many of these different microscopy techniques can fit together to be able to address different scientific questions. And cryo-electron tomography is one of those aspects, which is a really growing area for being able to answer questions at a different length scale in biology. So this figure, which was taken from a paper from 2020, really puts all of this within context. The amazing things that happen in dynamic imaging, in light microscopy, everything from super resolution, light sheet confocal technology, through to the really high level insights that you can reach with things like X-ray crystallography and single particle electron microscopy. In situ cryo-ET is almost like a new kid on the block that fits in between a lot of these different technologies. 15 years ago, this was really quite a difficult technique to invoke. There's a lot involved in being able to get that perfect cryo-ET image. And it's been a combination of technology development, expertise, and sample preparation that's really moving this now towards a context of being a standard technique within the whole facility. And it's really wonderful to see that transition taking place. So cryo-electron tomography, if you look at it in the context of volume EM, a fabulous talk from Andreas has really put all of this into context for us. Really, with volume EM now, you can answer so many questions on relatively large areas of biological material. Cryo-electron tomography answers questions at a different length scale. So down at the protein and molecular level and biomolecular structure level, you're able to reach sub-nanometer level insights. And it's this complementary data in combination with what you can achieve with the other volume EM techniques that allows you to be able to address new questions. And for today, it's the cryo side of tomography that I'm focusing on. You can also do TEM tomography on non-cryo specimens. Doesn't quite get you to the same level of resolution, but it's really the cryo stuff that I'm going to focus on today. And as I said, really, it's been the last 15 years where this technique has begun to have a real hold in the biological community. And if you look at the subcellular organelles that you can see with cryotomography, everything from mitochondria and the different subunits within the mitochondria, the endoplasmic reticulum. Looking at Golgi, this study here on the top right-hand side is the first one that was able to completely analyze Golgi apparatus ultrastructure under physiological conditions. You could look at lysosomes, you can look at cytoskeleton. So this is a cytoplasmic lattice in oocytes, and this was found to be composed of filaments that were also of maternal origin. And in the nucleus itself, looking at the kind of native chromatin fibers. And this is just a whole different length scale to what you could attain with any other kind of EM technique. So how does what does electron tomography data actually look like? On the left-hand side here, you've got your 3D data set that you capture from the tomogram. When we talk about 3D, when you're talking about electron tomography, you're talking about a slice thickness or a sample thickness of somewhere between usually 100 and 300 nanometers. So your actual sample is really quite thin relative to other techniques that you would use. But as a result, the resolution that you can get is much, much higher. So the nuclear pore complex you can see within this data, you can visualize what's going on in the cytoplasm. You have access to the nucleus and the components within that nucleus and the nuclear envelope. And it's biology at a length scale that really hasn't been achievable in any other way. You can then segment that as you would do any other data. Again, Andreas gave some fabulous examples from what you can do with Fib data in the last talk. You're able to, if you can identify what all these structures are, and that's one of the key things that's really exciting about this. Some of this stuff is actually new, but if you can identify what you're looking at, you can then segment it, and you can see this structural information within the context of the cellular environment. Which is very exciting. And all of this is in a completely unstained specimen. So this is cryo-imaging. You're freezing your specimen. You're not having to add any heavy metal staining to be able to get the contrast. It's all in native state. And it's an incredibly complementary technology to what you can do with other volume EM approaches. This is a unicellular green alga. It's used a lot to be able to kind of benchmark, I guess, some of the techniques for being able to look at the different subcellular components. But in terms of Cryo-ET, this is a FibSem, a plasma FibSem data set, and it gives you a fantastic overview of the cellular architecture. And if you look at all these different unicellular green algae, they all look pretty similar next to each other. So having this overall map from CryoVolume-EM allows you, once you get your really thin lamella from another algal sample inside your TEM, it allows you to navigate in a really sensible way to where you actually want to capture your tomography data set. So it gives you some kind of orientation and a map. So being able to see the same specimen at different length scales really acts as a guide for being able to reach those high resolution insights. And when you actually get your sample into the cryo-electron, when you capture cryo-electro tomography data sets from your sample, this is exactly the same specimen type. This is the algae again, but now using Cryo-ET. So now you get to a different length scale. You're not just looking at the overall apparatus or the nucleus or the basal body. You can actually see these in fine detail. You can see the mitochondria, the pyrenoid, the chloroplast, and now you can start to build up a picture at this really, really high resolution of what these subcellular components look like and how they all fit together. Excuse me. And as I alluded to at the beginning, it's not just about being able to see what we already know is there at higher resolution. That's already exciting, but the really cool thing that's coming now is that you're actually able to see biology that hasn't been visualized before. So on the left-hand side here, this is the same sample type. It's this alga where we've identified the cellular components for segmentation. But in the middle figure, what you're seeing in the red is the mitochondrion. What you're seeing in the kind of dark gray is the plasma membrane. You've got the microtubules, which are shown in pink. And what this data set showed is those yellow filaments, these filaments are actually tethering the microtubules to the mitochondrion. This is simply biology that hasn't been seen before. We haven't seen what these filaments are. We don't really know what the function is. There's lots more new biology that can come once you start looking at your samples at this new resolution scale. So being able to visualize, segment, and then actually explore what all these different cellular components are at this new length scale is helping to drive new biology, which is amazing. And it's not just unknown biology, it's also rare events. So because within your lamella or your specimen that you use for capturing these Cryo-ET data sets, you can see, relatively speaking, a largish field of view. You can see rare events, so things like mitochondrial fission within the ER membrane. You could look at these ciliary transition zones or these pyrenoid tubules and how they extend into the pyrenoid itself. So these rare events are visible, and again, all this is within the context of the cellular environment. So you're not extracting anything out to view it independently, you're actually seeing it within the context of the cell. You're also able to generate quantifiable insights. So we're not just looking at, it's amazing to see the cells at this kind of resolution level, but you're not just looking at individual events. You can actually start to generate quantifiable insights as well. As shown here, this is a slime mold specimen, and this group were interested in an actin wave moving through the specimen. So they stimulated the specimens, they preserved them, and then imaged them using Cryo-ET. And they were able to identify the filamentous structures, which is the actin. You were able to segment that out. This was all done with the Amira software. And you're able then to start looking at different things. You can look at directionality, you can look at number, you can look at length. And they were able to quantify, in this case, after the segmentation, you've got all that parameter space available to you. So in this particular case, the polar angle of the filaments has been color-coded, and you can see exactly where the actin wave is. Actually, the blue is shown for the actin wave. You can also color code in a different way. So this time it's looking at the azimuthal angle, and this highlights the direction of the actin polymerization. And this gives possibilities to be able to group these two different groups together to look at different characteristics within the cell. So we're moving from having these individual, really amazing insights of new biology at a length scale that we haven't seen before into quantifiable insights, and even comparing different groups, so different genetic models or different disease groups. We're now at the stage of being able to scale tomography data to be able to answer new biological questions, which is really very exciting. So what kind of samples can you image with Cryo-ET? You've got everything from proteins and viruses through to bacteria. You can image cells, so these are cells on a grid. You can also then start to move into 3D culture. And again, Andreas in the last talk gave a great example of why this is important. Biology doesn't just happen in 2 dimensions or even in a single cell culture. Also looking at the interactions between multiple different cell types in a 3D environment is important. And then of course, scaling up to bulk tissue and small organisms. So when kind of tomography first came about, being able to even image proteins, viruses, and the really thin parts of cells was incredibly exciting. But what we're seeing now is that a lot of the work is moving to the right-hand side of this graphic, where we want to be able to capture what's happening from a 3D specimen point of view. So in these organoids, in these bulk tissues. And this requires slightly different technology. So to be able to generate a specimen that's suitable for tomography, it needs to be thin. It needs to be ideally within this 100 to 300 nanometer size. And if you have your sample is much thicker than that, you need to be able to generate that. That's that lamella, we call it. And that is done using a cryo-fib milling system. So you mill either side of your specimen to generate your lamella. You also have considerations about how you freeze your specimen. So as the sample gets bigger, you need to start using high pressure freezing techniques. So the complexity of the experiment increases as you move to the right, but the possibilities for what the experiments can generate also increases. And that's really where we see a lot of our user base starting to increase the usage in tissues, bulk tissues and organoids. So from the cryo-fib perspective, this is exactly how it works. So you fib mill your specimen from either orientation to be able to generate this lamella, which is somewhere between 100 and 300 nanometers. And then you take that lamella and you put it into, to be able to generate that lamella, you can do it with a couple of different systems. So we have a gallium fib system, and we have 2 alternative plasma fib systems, which are routinely used for being able to generate cryo-lamella. The Aquilos 2 is our gallium fib system. It's incredibly dependable. It's really proven itself to be a workhorse for being able to generate lamellae. More recently, plasma fib technology has kind of hit the headlines, and we have 2 flavors of plasma fib. The Hydro-Bio, which is a fantastically adaptable machine. You can use this thing for so many different options and experiments, including volume EM and lamella prep. And the Arctis, which is really designed to allow you to scale your lamella prep. So higher throughput production of lamellae for cryo-TEM. I'm not going to talk more about these today. I am going to enthusiastically plug a webinar that we have coming out next Tuesday, plasma fib technology and how it supports cryo-lamella prep and volume EM. We've got a lot of different users who are already making use of plasma fib, talking about their experiences. Please do come along, join, ask questions about how they've got on with being able to use this technology, because it's exciting to see this workspace moving forward. So once you've captured your lamella, or you've generated your lamella, so on the right hand side here, what you see is a lamella actually in preparation. So this is again this algae being created, being prepared in lamella form to be able to go into the TEM. You then move your lamella into your cryo-TEM. And we have a couple of different versions of cryo-TEM, which are also suitable for being able to capture this data. We have the 200 kV Glacios 2, and we have the 300 kV Cryos. Both are fantastic for being able to make really beautiful tomography data. And it's been a journey to be able to get there. It's not easy to be able to generate instruments to create dependably really good data, and what you're seeing here is the endpoint of a lot of hard work, tweaks, working with users to get the best possible instruments to be able to generate these incredible tomographic images. So if you want to find out more, visit the website or speak to anybody from the Thermo Fisher team who'd be able to help you think about which instrument might be best, which workflow might work best for your particular specimens. So if we go back here, we've collected our focused ion beam milled lamella, but there are a couple of things about that lamella that we really need to make sure are optimal before we move over to the TEM. We need to make sure that the lamella that you generate are really, really of high quality. It's the same with any imaging technique. If your lamella are not good quality, you're not going to get great data out of the end of it. And also, and what we're seeing more and more, it's not just about having that one perfect data set. You need many, many different data sets to be able to generate a representative overview of biology. And both of these things kind of work in parallel to be able to really push the technology space and the applications forward. So if we look at the quality side of this, being able to make sure that what you're interested in is actually in your lamella is very important. And fluorescence tagging is a way of being able to do that. You know where your protein is because it's either got a fluorescent protein attached or you've got an antibody in there, and you can use correlative techniques to be able to identify exactly where that protein is in your three-dimensional specimen. You can then target the lamella preparation region based around that fluorescence. Now, all of this in the Thermo Fisher systems happens in the same instrument. So inside our FIB systems, you can have a integrated fluorescence light microscope, the IFLM. All happens within the same software, so it makes it very easy to be able to identify the region that you want to capture inside your lamella and then set off the FIB milling process to be able to make sure that your protein or your molecules of interest are actually in the region that you generate the lamella in. So that really helps with the quality and reduces the number of times that you don't actually capture the protein within the region that you want. And from the other point of view, and this really is kind of hot off the press in a way, being able to generate multiple lamella in a dependable way that allows you to be able to scale up your experiments. As I said, one data set already is amazing in terms of tomography. And 15 years ago, it was an incredible achievement to get to the point where you had that data set. It was really hard work to get there. Now things are changing. The technology has improved, the sample preparation techniques have improved, and the capabilities like the Arctis, which is our high throughput FIB system, are now there to allow you to be able to scale up to multiple lamellae. So now you can start to really generate quantifiable, reliable insights into your specimen. And this paper, which is still preprint at the moment, really is kind of a step towards showing how that can have an impact from the biological perspective. They captured 1,829 raw and processed tomograms of the single cell alga. And these have all been deposited, actually, again, talking to the last talk, into the Empire database. So the idea now of having this compendium, which is publicly available, data sets that allow you to be able to dig in and have a look and explore this incredibly rich resource for being able to drive method development and inspire biological discovery. And I think this really sets the benchmark for what's going to happen moving forward. We need one of these central resources for many, many different cell types and different tissue types, different disease groups, for example, that allow other biologists to come in and ask different questions of the same data. Equally, all of the reconstructed tomograms from this paper have been deposited in the Chan Zuckerberg Cryo-ET data portal. So there's a huge amount of resource out there. Go and check it out, have a look, see if there's anything in here that you can actually use to be able to address your own questions, or if you run a facility, point users towards this resource. It really, I think, is setting the standard for what's going to happen now in terms of being able to generate these libraries of incredibly rich data. And I love the quote from the paper: "We invite scientists, educators and students to interactively explore these tomographic volumes and accompanying annotations of the native cellular environment." And it's going to be exciting to watch as this and other similar resources really set the stage for being able to dig more biology from these things. So within this same paper, there were many different aspects of cell biology that were pulled out: microtubules, clathrin, ATP synthase, rubisco. And the kind of idea, or one of the visions really of the paper, was that different groups could dig in and start to look at different proteins or protein complexes or mixtures of proteins within the specimen. And that has actually already happened. From the respirosome point of view, there was a beautiful science paper, which I have to say has one of the most amazing science cover graphics that I've seen. Really beautiful stuff. They used this Empire resource of data to be able to look at the native structure and the molecular organization of several different mitochondrial complexes. And this has provided new clues for the functional significance of these respirosomes, which are these super complexes. And nobody really knows exactly why these super complexes exist. They seem to be pretty stable. They always seem to be present, but nobody really knows why. And this is the first step to really understanding the functional significance of these. And it also provides the blueprint in this paper for others who might want to disrupt the supercomplex. There's now a benchmark for what the supercomplex looks like. Now let's look at what happens when you disrupt it and how that impacts on how the different units of the super complex all fit together. So yeah, very exciting science. And I couldn't not show one of the amazing movies from this paper. So this is one of the tomograms that they used. They were able to segment out these different components of the respiratory chain and really explore within the cellular context how these aspects were sitting next to each other at a length scale that just hasn't been feasible before. And really this central database of all these different tomograms is really going to help drive similar papers, but asking completely different questions of the same data sets. So yeah, very exciting times. So what's the role of the core facility in all of this? I have experience myself of going to conferences and seeing really fantastic presentations and being inspired by what technology can provide, come back very excitedly, ready to start my experiment, and actually have no idea how to get going. I don't know how to start. I don't know what I need to do to my sample. I don't know how big my sample needs to be, all these questions. And similarly to many other imaging techniques, kind of what you see at the conferences is the end point of a lot of hard work to be able to get there. And every step of the Cryo-ET pipeline requires optimization. You require, in some respects, different instrumentation. You require different expertise to be able to get the best and the most out of your samples. And really, as a central facility, the provision of access to this complete workflow is one key element. Being able to have all of the high pressure freezing if you need it equipment, and the sample prep equipment, everything that you need around lamella prep, so the cryo-lamella preparation instruments, and then of course data collection on your cryo-TEMs and the glacios on the cryos. All of that's very important, but having the kit is half challenge. You then need to be able to optimize every single step, and being that kind of go-to place is really what's allowing this high throughput production of data that we're now seeing coming through the literature. So providing expertise on sample prep, optimizing acquisition parameters and conditions, clearly that's important. And almost more importantly, I would say, is guidance for users to generate a realistic experimental plan. So expecting to be able to see sub-nanometer resolution in a sample the size of a football, clearly that's not going to work. And I think being able to sit down with users and come up with a realistic plan is one of the most important things that the facility can offer alongside their expertise. And also collaborating with other facilities. This isn't a workflow that needs to necessarily happen all in the same place. There could be people who have sample prep technologies in one place. There might be a FIB system somewhere else, and perhaps a cryos or a glacios somewhere else. So being able to match together where all of these different parts or pieces and parts of the workflow go is also really important. And I see Euro-BioImaging having a really important role there to be able to match together users who come back from conferences super inspired with ideas, to match them with kind of which facility can really help direct to be able to get to these really amazing results. So there are many different applications, and I hope I've at least inspired you a bit in terms of what possibilities there are in the realm of cell biology for tomography. For me, as a light microscopist by trade, I think it's incredible how this complements what we can do in light microscopy and in the rest of the volume EM techniques and technologies. And it's moving quickly. As I said, 15 years ago, this stuff was really, really hard. Being able to produce a lamella and get a good image using a cryo-TEM system was really, really difficult. Now it's becoming much more streamlined and therefore much more accessible for the multitude of users who would be interested to do it. And we're seeing the high level publications come to a penny actually in great high impact journals, because it's just such new biology. So yeah, that was it for me on the cryotomography side of things. I would just like to have a couple of plugs for events that we have coming up. We have this webinar next Tuesday. Please do come along. If plasma fib or fib technology piques your interest, or you'd like to learn more about what it does, please come along, hear from these scientists who've really doing amazing things with the technology. It should be a really good day. And on the 17th of July, we have another. So Jirka and Nicole are going to be talking about serial block face. In some respects, their kind of journey, but around being able to optimize imaging to be able to do the really hard stuff. So getting the best results from your specimens, being able to image very large volumes, both with tile scanning and also over weeks and weeks of acquisition. So there's QR codes for both of those. Please come along and join us. And I'd just like to say thank you for the opportunity to present, and I'd be glad to take any questions.